The broadcast is now starting. All attendees are in listen-only mode.
Hi, everybody. My name is Andrew Olson, and I want to thank you today for attending our webinar. Today's presentation, Dr. Sidra Ahsan, who is the product manager for iPathway Guide, will walk you through a comparison of protein and mRNA expression profiles. Dr. Ahsan's areas of expertise include molecular biology, cancer genetics, pharmacology, and stem cells. She also has authored several peer-reviewed publications on the topics of targeted therapeutics and variants in cancer. In today's presentation, she'll walk you through a data set where she will compare mRNA and protein expression signatures. As you may know, Advaita Bioinformatics has a cooperative relationship with SCIEX, a leader in mass spectrometry proteomics. And if you are a SCIEX customer, I encourage you to speak with your SCIEX sales representative about special pricing and packages that we have for iPathway Guide through SCIEX. As we go through today's presentation, please use the question window in your webinar control panel to ask any that you may have. At the end of the presentation, we will address as many of these questions as we can. But if we don't get to your question during the webinar, we will be sure to follow up with you directly afterwards. A recording of today's webinar will also be available. You will get a link either automatically through the webinar system, or it'll also be available on our website in the webinar section of the support area for iPathway Guide. So with all of that taken care of, I'd like to introduce Dr. Sidra Ahsan, who will walk us through today's presentation. Sidra?
Thank you, Andrew, and welcome everybody to the fourth webinar of our 4-webinar series. If you guys have attended the previous webinars or are aware of it, the previous 3 webinars we concentrated on mRNA expression data. So I really wanted to talk about protein data in this webinar. And the agenda for today's webinar is that I'm going to first briefly introduce you to proteomics and go over the proteomics bioinformatics pipeline. I will show you how to upload proteomic data in iPathway Guide and how you can generate a meta-analysis report to compare your protein and mRNA expression data. This is a pretty hot topic nowadays, comparing protein and mRNA expression data, because obviously mRNA expression does not always necessarily translate into protein expression. Then I will dive into a case study in which I'll be comparing both protein and expression, mRNA expression data, and I will show you how you can identify common molecular mechanisms between the 2 data types, how you can identify some biomarkers, and also how you can identify drugs to target a specific biomarker that you're interested in, after which we'll have a brief Q&A session as well. And feel free to type in any questions that you have during the presentation as I go along in the chat box. So proteomics is defined as the systematic high-throughput approach to protein expression analysis of a cell or an organism. And it's pretty much analogous to an mRNA expression study, just with proteins. And you can extract many different types of information from proteomics, such as the abundance of proteins in a specific sample, structural information about proteins, such as variations and modifications, as well as protein-protein interactions. In this webinar, I'll be concentrating on analysis of protein abundance, which is also called expression proteomics, which is defined as the quantitative study of protein expression between samples that differ by certain variables. So as I mentioned, that this is very analogous to mRNA expression studies, where you have 2 different samples that you want to analyze for different expression, different mRNA expression. You can do the same exact thing with protein expression. So briefly, I wanted to introduce everybody to the proteomics workflow, because I'm not sure what exactly the background is of all the attendees, and it is a relatively new workflow. So you start out with a biological question, and you have cells and tissues from which you extract proteins, And then these proteins are digested with a protease or a combination of proteases into peptides. And these peptides are then sprayed onto a liquid chromatographer coupled with a mass spectrometry machine. And there are really two approaches for this type of sequences, two of the most common approaches. One is shotgun sequencing that provides a deep coverage, and then you have the targeted MS sequencing, which provides more quantifiable information about a specific set of proteins. And what happens in the mass spec machine is that the mass to charge ratio of a protein and the fragmentation spectra is recorded. This mass spec data, which includes peptides, is then aligned to a sequence database, after which it is assembled into proteins and quantified, and a differential expression file is produced if you are comparing two different sample types. And then from this differential expression file, you can do further functional analysis, such as pathway analysis, gene ontology term analysis, in bioinformatic applications such as iPathway Guide, and that's what I'll show you today. So there are two different types of file formats that are accepted by iPathway Guide for proteomics data. The first one is the SCIEX SWAT 2.0, and the second one is custom tab delimited file. As Andrew went over in the introduction, that we have a cooperative relationship with SCIEX SWAT, so you can directly upload data coming from any SCIEX SWAT instrument. And next I'm going to go into the pipeline that you will use to do that. So using a SCIEX SWAT spectrometer, after you have gotten your peptide information from there, you can use many different applications, and I have one of the more common workflows listed here, such as the protein expression extractor and the protein expression assembler, and these, I believe, are available on BaseSpace. And what happens with the protein expression extractor is that it extracts and integrates the peptide or protein peaks using the ion library, and then the assembler normalizes the data and computes the protein fold changes across these samples. And this data can be directly then uploaded into iPathway Guide. If you are not using a SCIEX instrument for mass spec, you can still analyze proteomic data into iPathway Guide using the custom tab delimited file. And this is very similar to what we require for mRNA expression data, in which you need gene symbols, log fold change, and associated p-values. What is important here is to remember that you need to convert your protein IDs into gene symbols, because that is the required input of iPathway Guide. And there are many commercially available applications free that will do that for you. I'm not going to go over and show you how you can upload the custom tab delimited file or the SCIEX file into iPathway Guide, because it is the same procedure for the mRNA data as well, and we have gone through that in our previous 3 webinars. But just to provide you with the snapshot, that this is the intake form in which you can choose your organism, the file format, and then upload your file. For custom tab delimited file, you'll have to select the columns for your gene symbol, log fold change, and p-value, and you can easily upload your file into iPathway Guide. So now I'm going to dive into the case study. And the goal of this case study is to compare the protein and mRNA expression profile of metastatic cancer cells. And the way I did this is that I first analyzed protein expression data, case versus control, and I then analyzed mRNA expression data, case versus control, coming from the same cells. And then I compared both this mRNA expression and protein expression data through the meta-analysis feature in iPathway Guide. And I'll show you how I did that. So for the proteomics data, as I said, I wanted to concentrate more on the protein data in this webinar. This data was obtained from a paper that was recently published in Scientific Reports, which is a Nature journal, And in this paper, what the researchers did is that they compared an epithelial bladder cancer cell line versus a metastatic bladder cancer cell line. These cell lines were T24 versus T24M. T24M is a metastatic bladder cancer cell line, which was derived from T24, so it was a syngeneic study. And after choosing the log fold change thresholds for 0.4 and p-value of 0.05, I was left with 356 differentially expressed proteins out of a total of 530 proteins that were measured in this study. And I obtained all of this information from the supplemental data published in this paper. So next I'm going to show you how you can generate the meta-analysis report. As I said earlier, that we have gone through how you can analyze a single contrast in iPathway Guide, and proteomic contrasts are analyzed the same exact way as transcriptomic data. So I'm not going to show you that, but I'll show you how we can create a new meta report in which you can compare the protein and the mRNA expression data. I have listed the steps here on how you can do this, but I'm going to show this to you within the application. So here's the dashboard for iPathway Guide, and if you are familiar with the application or you've seen the previous webinars, I'm sure you have seen this before. You are going to click on the plus sign here which says Create New Meta Report, and you will give your report a brief title and a brief description, after which you will select the single contrast that you want to include in this report. So it's important to remember that the proteomic data was analyzed on its own, case versus control, and mRNA expression data was also analyzed, case versus control, and now I'm going to generate the meta-analysis report of the two. So I'm going to click this here and add this report to my selected contrast for the meta-analysis report, just the total mRNA expression one. And now I have added the selected contrast that I want to look at, and then I can click on Create Report here. It takes anywhere from a minute to 2 or 3 minutes for this report to generate, and you can see the status for that in the dashboard. And you'll also receive an email as soon as the meta-analysis report is created. So now let's take a look at this meta-analysis report. I already created the report previously, so I'm going to show you the report here. So the default page for meta-analysis is the genes view, and you can see the layout here where you have a Venn diagram which shows you the logical relationships between the contrasts that you're looking at, and followed by a table down here that you can easily search or view in the form of a rank diagram. And on the right side of the page, there will be further details about what you have selected here, such as a gene pathway or a gene ontology term. So in the pink, we have the mRNA expression data, and in green, we have the protein expression. And it's really interesting because it seems like there's almost an 80% overlap between the two data types of the genes. However, these 213 genes does not mean that their expression change is the same between the protein and the mRNA expression, but that between the protein and mRNA expression, these genes were common. These genes and proteins, I should say, were common. So let's say I'm interested in looking at these 213 genes. I can easily click on this, and this table down here below is going to show me these 213 genes that are common amongst the two different data types. I can also view this in the form of a rank diagram. I want to look at everything from the perspective of proteins, as I mentioned, because that is my primary case study at this point. So I can easily look here and see which are the top 20 proteins and what their rank is compared to the mRNA expression. And one of the proteins that jumped out to me was the STAT1 protein, because I did a little bit of research on bladder cancer and found that this is one of the most highly expressed proteins in bladder cancer and has been widely used as a therapeutic target for metastatic bladder cancer. And now I can quickly also obtain some more information about this protein in the summary here on the right side, and I can look at the expression change by contrast in both the data types. And it's interesting because at the level of transcript, STAT1 is upregulated. However, at the level of protein, it is far more upregulated. It's about there's about a threefold difference in the expression of protein and mRNA, which leads me to believe that most likely there is some kind of a post-translational mechanism that is going on here where there is more stabilization of the mRNA or something of so forth. So really quickly, I was able to, generate a hypothesis that I can further study in lab, that if I needed to look at, if I wanted to look at why the mRNA levels don't exactly correlate with protein levels, even though they're both upregulated. Let me quickly go back to the slideshow here. So just to review, using the meta-analysis feature in iPathway Guide, I looked at the common genes and proteins between the protein data set and the mRNA data set. And one thing that I might have forgotten to mention, which is important to know, is that these cells were the same exact cells from which the protein and mRNA were extracted. And what we found was that about 80% of genes are at least translated into protein in this data set. And then I wanted to concentrate on, I wanted to look at STAT1 because this is a very important cytokine and it's known to be implicated in the advancement of bladder cancer cases. So I found some more information about STAT1 within the genes view of meta-analysis, and this summary actually comes straight from the summary that's provided in iPathway Guide. There's also an NCBI link in which you can get more information about the gene, and I've also listed the citation here, which shows that these STAT1 levels have been shown to be very high in metastatic and advanced bladder cancer cases. So now I wanted to answer some specific questions about metastatic bladder cancer using the mRNA and protein expression data. So one of the first questions I had is that what are the common molecular pathways and biological processes based on mRNA and protein expression between metastatic bladder cancer and non-metastatic bladder cancer cells? So let's go back to the analysis. So now I'm going to answer these questions, I'm going to first look at the pathways that are common or unique between the proteomic and the transcriptomic data set. So this was really interesting to me that even though there are so many genes that are genes and proteins that are common amongst the two different types of data sets, there's only one pathway that came up to be significantly common amongst the protein expression and the mRNA expression data set. And this one pathway was the cytokine receptor interaction pathway. And if you look at the pathway here, it's fairly interesting because most of the genes in this pathway follow the same change in expression. So if the mRNA is downregulated, the protein is downregulated as well. Now let's take a look at all of these pathways, because there aren't many significant pathways since we just started out with 356 proteins and there were about 350 differentially expressed genes in the data set. So I'm going to look at this in the form of a rank diagram and from the perspective of proteins. And metabolic pathways is the most significant pathway in the proteomic data set. However, it wasn't significant in the mRNA expression data set. And also pathways in cancer, small cell lung cancer pathway was very significant in the protein data set, but not significant in the mRNA expression data set. So let's see what's going on from the side of the mRNA expression. And where you see a lot of cytokine receptor interaction pathways, here's the TNF signaling pathway, which is also sort of related to cytokines and chemokines. So there are differences in signaling pathways that are significant between protein expression and mRNA expression, even though a lot of the genes and the proteins were similar. Now I wanted to look at the biological processes amongst the two. So let's look at gene ontology terms. And here you have a lot more biological processes that are common amongst these two different data sets. I'm going to apply a correction factor here. And we've gone over these correction factors in the past, but I just wanted to reiterate that for gene ontology terms, I highly, highly recommend you use one of the pruning correction factors because they really help reduce the noise that you get with the gene ontology term. So I'm going to select Elim pruning here. After Elim pruning, it seems that there are 5 biological processes that are significant and common amongst these two data sets. So let's look at what these 5 biological processes are. And I'm going to look at this also in the form of a ring diagram. So this was really interesting to me because even though metabolic pathways was not significant signaling pathway in the mRNA expression data, if you look at the 5 common biological terms, you'll quickly see that they're either related to metabolism or they are related to angiogenesis and metastasis, which makes sense because we are comparing a metastatic bladder cancer cell line to a non-metastatic bladder cancer cell line. And so you would expect metabolism to play a role, as well as some of the metastatic properties such as angiogenesis and blood vessel endothelial cell migration to play a role in this. So I wanted to look at response to drugs because this is the top most common pathway amongst the data sets. And I can quickly on the right side look at the differentially expressed annotated genes to this biological term. And you can see here that IL-6, which is a very well-known cytokine and is known to be present in a lot of different types of advanced cancers. And it's actually a very good biomarker because you can detect it directly from blood and is upregulated on the protein level as well as the mRNA level. And then you also have some STAT1 here, which is also annotated to response to drugs, which I just went over, and as well as TGF-beta1. What is interesting about TGF-beta1 here is that the mRNA is downregulated, however, the protein expression of TGF-beta is upregulated. So this is also something that might be of interest to someone if they wanted to do a mechanistic study to study why mRNA expression is down and protein expression is up specifically for TGF-beta. And then you also see VEGF-C here, which is also another important protein in angiogenesis and metastasis as well. So I kind of have an idea of, some of the biomarkers for some of the biomarkers that I could possibly follow for this metastatic bladder cancer because I have STAT1 here, there is IL-6 here in which both the mRNA and protein levels are up. And if I wanted to do a mechanistic study, I could look at this TGF-beta1. Before I proceed to the next thing, I want to just review what I have gone over. So one of the common signaling pathways in between the protein and the mRNA expression data was the cytokine receptor pathway. And interestingly, IL-6, TGF-beta1, which are both parts of the pathway, were present in both on the mRNA level as well as the protein level. And IL-6 could be used as a potential biomarker because you see an increase in the IL-6 transcript as well as the IL-6 protein. So you can target it as well with a drug on a protein level or molecularly on the mRNA level with an siRNA or also a microRNA if you were interested in that type of a study. And then TGF-beta1 was something that I thought would be interesting if for a mechanistic study because you see that the RNA for TGF-beta is downregulated whereas the protein is upregulated, and this could be a possible mRNA stability issue where the mRNA is being degraded way rapidly, and And actually, the mRNA would not be degraded, right? Even though even though not much of the mRNA is being produced, the mRNA is not being degraded rapidly. So you're seeing still a lot of increase in protein expression, even though the mRNA is downregulated. And next, I looked at the gene ontology terms, in which I found response to drug to be the #1 most common biological process amongst these two different data types. And interestingly, all of the common biological processes were related to metabolism and metastasis. And if you remember, in the protein data, the metabolic pathway was the most significant pathway. So there is a concordance between the analysis, the gene ontology analysis, and the pathway analysis. And in response to drugs, and in looking at the response to drug biological process, the genes that were annotated to it, some of the same genes that we saw in the cytokine receptor interaction pathway came up, such as IL-6, STAT1, and TGF-beta1. So this gives me more confidence to possibly pursue some studies on IL-6 or STAT1 or TGF-beta1. So let me actually go back to the application, because I wanted to show you how, suppose you were interested in targeting IL-6 because it is upregulated in the level of mRNA and protein. So how would you exactly go about targeting that? Where can you find more information? Instead of having to go outside of the application, you can actually find that information within the application. So with the meta-analysis tab in iPathway Guide, you also have each one of the single contrasts available right there, that if you wanted to switch back and look at a specific pathway, you could. So I'm going to click on this T24M, the proteomic data set, the single contrast between the metastatic and the non-metastatic bladder cancer cell line. I can quickly go to the genes page here, where it shows me all of the genes that were measured and were significant in this data set. I can type in my gene of interest, which is IL-6. When I click on IL-6, I have a lot more information about this gene present here, such as the biological processes, molecular functions, and cellular components it's annotated to, as well as the pathways this gene is involved in, and also the drugs that are known to target the protein and the microRNAs that regulate this gene as well. So let's look at the cytokine receptor interaction pathway, because this was the common pathway between the protein and the mRNA expression data set, and IL-6 is a major component of this pathway. I have gone through this whole pathway page in different scenarios in the previous webinar, so I'm not going to concentrate on going through every detail here, but what I wanted to show you is that you have a pathway map available right here, and in the pathway details view, you can look easily at the list of approved drugs for this pathway, as well as the microRNAs that target or regulate genes within genes or proteins in this pathway. Because I'm interested in IL-6, I can quickly click on IL-6 here on the pathway map, and now I only have a list of drugs that target IL-6 and the list of microRNAs that regulate IL-6. So there are two drugs that came up here, siltuximab and sirukumab, which are both monoclonal antibodies, as I can see by their name. And if I click on any one of these drugs, it'll give me more information about the specific drug from an outside source, the CAG drug database. And let me go back to the pathway details here. And if I check the box next to the drug, it'll show me the targets of that drug. So you want to make sure that you're using a drug that only has one target and not multiple targets because of potential side effects. And then I'm going to do the same for sirukumab. And I can see here that both these monoclonal antibodies are specific for IL-6. I can also, if I wanted to target IL-6 mRNA instead of the protein, I can look at or study some of the microRNAs that are known to regulate IL-6. And LET7A and actually the LET7 family are some of the most common family of microRNAs that are implicated in cancer subtypes. So it makes sense that a lot of the LET7 microRNAs regulate IL-6. It is a very well-known cytokine. So really quickly, I was able to obtain a lot of information from the same exact application. Instead of having to go to any outside data sources, I not only identified IL-6 as a therapeutic target, because both mRNA and protein levels are high for IL-6, but I also showed you how you can target IL-6 on 2 different levels: protein via drugs, and then the mRNA via the microRNA. So you can modulate the microRNA to target the mRNA of IL-6. So this is a quick summary of the slide where you have the pathway map with IL-6 highlighted and shown as one of the targets, and you can target IL-6 with the 2 drugs that I mentioned. So in summary, I have gone over the proteomics bioinformatics pipeline with you, where I showed you how you design, or how does a proteomic study come about starting from a cell in a tissue. And I showed you how you can upload protein expression data into iPathway Guide. We accept 2 file formats, the SciEx SWAT file format, as well as the custom tab delimited text file for any other instrument that is not a SciEx SWAT instrument. And I also showed you how you can generate a meta-analysis report comparing both protein and mRNA expression data. And then in the case study in which I went over the comparison of a metastatic bladder cancer cell line versus a non-metastatic bladder cancer cell line, I compared the expression, the mRNA expression with the protein expression to show the common molecular mechanisms between the protein and mRNA expression that are listed here, the common signaling pathway, as well as the biological process. And then I was also able to identify a plausible biomarker for metastatic bladder cancer. And my focus here was that I was looking for something that had both mRNA and protein expression that was upregulated, not just one or the other. And then I also showed you a plausible therapeutic strategy by targeting IL-6 using one of the 2 monoclonal antibodies. And the information of that, all of that, I found really easily within the application. I wanted to keep this webinar pretty basic because this is a newer topic. The proteomic analysis itself is a newer field, and then the comparison of the 2 protein and mRNA expression is even a newer field, is even more new than the proteomic studies. So if you are obviously interested, you can actually compare 2 protein samples versus 2 RNA samples. I just did a 1-to-1 comparison, but obviously you can, because you can compare and contrast up to 5 different experiments in the meta-analysis feature of iPathway Guide, you can go up to 5 different types of data sets, protein or mRNA data sets, and compare that within the meta-analysis feature. And with that, I will take any questions. And since everybody, we're going to wait for some of the questions to come in, I wanted to let people know that we will be having our next webinar on June 28th at 2 p.m. on variant analysis. Advaita Bioinformatics has a really cool application out, iVariant Guide, which performs variant analysis, and it does it better than most other applications that are out there. And I highly recommend you attend this webinar, which is on, June 28th.
Great. All right. Thank you. Wonderful job. We do have a number of questions, and let's just kind of start. We'll just kind of go through them in chronological order here.
Okay.
So the first one that we have was pretty basic. How many replicates were analyzed in each data set?
So as I said, that I took this information from the supplementary data of a published paper. I'm going by what they said in their paper. They said that they had 3 replicates for each data set.
Gotcha. Okay, thank you. And another question here, when we talk about log fold change, are we referring to log 2 or log 10?
So in this webinar, I was referring to log 2 fold change. You can upload you can upload data with log 10 fold change if you would like. Whenever you when you upload your file in the reports intake page, you can but just know that choose your thresholds accordingly. So we have these default thresholds set for log fold change of 0.6 and the p-value at 0.05, and the log fold change of 0.6 is corresponds with log 2 fold change.
Gotcha. All right, very good. So we had another question here. Do the overlaps in the Venn diagram, when we're looking at the gene level, does that only include proteins and genes in which the change is in the same direction, or does it also include those significant genes where the change may be in opposite? So it's like the gene is up and the protein is down. That's
actually a really good question, and I think you worded the question better than I had explained the answer during the webinar, but it only includes the common genes and proteins. It does not mean the same direction. So you can use the sortable table below to look at the direction, and it's easy to look at that because everything that is upregulated is in red, and then the downregulated genes and proteins are in blue, so you can easily look across the table and see, you know, which genes follow the same trend. In this specific data set, most of the genes and proteins followed the same trend. However, there were a few where you would see mRNA up and protein down, or vice versa.
Mm-hmm. So this may be a little bit broad, but so the question is, how are these comparisons being made? And I think this is when we're looking at the pathway and the biological process analysis. Are both controlled data sets and contrast data sets combined, or is there a set of metrics generated for the mRNA and protein data set that are being compared?
Both data sets are being combined.
But they're analyzed independently?
Oh, right. So each data set is analyzed independently. Each data set has its case versus control. The mRNA data has been analyzed and metrics have been generated for that. So I guess that's how, that'll probably explain it well, is that for each of the single contrasts, let's say mRNA data, case versus control, the metrics are generated. For protein data, case versus control, metrics are generated. In the meta-analysis then, these are combined. However, also, as I showed you in gene ontology terms, you have these correction factors available. So you can use those correction factors in the meta-analysis and correct for p-values for multiple comparisons.
Okay, great. Next question, can I get a copy of the webinar, please?
Yes, yes. I will make sure. I'm not sure if I posted the slide deck, but I'll make sure to post the slide deck, and I'll write your information down.
Well, and not only that, the webinar will also be is being recorded and will be made available to everybody. You'll get a link with. You will receive a link to the recording within the next 24 hours. So look out for that. All right, great. Next question was, are there any plans to expand the number of comparisons that you can make in the meta-analysis?
So we've had that question from clients before as well, and it's because the Venn diagram gets very busy, which is the reason why we have limited it up to 5 comparisons. However, it is a possibility. We are looking into increasing the number of comparisons for meta-analysis in the near future.
Next comment or question, interesting webinar. Is it possible to integrate microRNA expression data in the presented analysis?
So I actually did not really touch on microRNA expression in this webinar because I didn't want to jump from mRNA and protein and microRNAs, but we at this time do not accept microRNA data. We only accept mRNA and protein data in iPathway Guide. And however, if in the near future we do decide to include microRNA data, this would be something that we'll definitely include in the meta-analysis portion.
All right, great, thank you. Another question, is it possible to look at the interactions between the common and differentially expressed proteins and the genes in pathways in iPathway Guide?
So I believe you're talking about, so you can obviously look at them in the pathway map that I showed you towards the end of the single contrast. You can look at the single contrast and the pathway map. However, if you are asking for the interactions of the protein-protein interactions and generating your own analysis, this is also something that we have just recently talked about and will be available in iPathway Guide hopefully by the end of this year. But using the pathway map, you can at least get an idea of the gene and the protein interactions that are standard and well known. All
right, and then I think this will be our last question here. did you use the same p-value cutoffs for both the proteomics and the mRNA expression data sets?
So for log fold change cutoff, I used the same exact. However, one of the problems that I did run into in the gene expression data was that the p-value wasn't provided. However, I do highly, highly recommend that you use the same exact threshold, the log fold change and the p-value thresholds, when you're doing a meta-analysis, just to have a cleaner analysis, of course. But in this data set, I ran into the problem that they did not provide p-values for the RNA expression, so I did assign an arbitrary p-value to the log fold changes, which are the most important part for our impact analysis. The p-value of 1. The arbitrary p-value was chosen as 1 for the gene expression data set, but as I said, I highly recommend choosing the same log fold change and p-value cutoff threshold if you're going to do meta-analysis.
Okay, great. Well, with that last question, I think we'll wrap up today's webinar. If we did not get to your question, I do apologize. We will follow up with you individually afterwards, and we'll be sure to address your questions personally with you. I encourage you to use the application and check out our new application, iVariant Guide, and attend our future webinar. You will get a reminder to sign up for that in the future, and we hope to see you again on June 28th. Thanks, everybody. Have a great day.
Thank you.